All right, and we go live.
Devansh, you can have your video switched on.
Yes.
Initially, yes, please. Yeah.
Yes, a very good afternoon, all the attendees. Yes, a very good afternoon to all the attendees. It's a pleasant day indeed, and in continuation of our Sharing the Learning series on webinars, we are pleased to bring to you an exciting webinar on a very hot topic in today's world. So, as the world, there's a in this world, there's a mad rush for vaccines to develop vaccines against this pandemic, and biopharma technologies and Spinco Biotech have developed solutions to support you for complete freeze drying of all vaccines and diagnostic products. Today, we are pleased to have Dr. Kevin Ward, Director, Research and Development, Biopharma Group, United Kingdom, Sally Potentier, Sales Development and Courses Executive, Biopharma Group, UK, Clara F., International Business Development Executive, Biopharma Group, UK, Dr. Uma Raghuram, Associate Vice President, Biopharma and the Country Head for Biopharma, Spinco Biotech, and Mr. Mohan S., the Country Head for the Process Technology Division. I would like to request Dr. Uma Raghuram to begin the session with some words of wisdom.
I'll just share my screen first. Very good afternoon to you all. It is indeed our pleasure to welcome all our guests on this webinar on freeze drying of vaccines and diagnostic kits. We are very pleased to have with us Dr. Kevin Ward, Ms. Sally Potentier, and Ms. Clara of Biopharma UK. I take this opportunity to briefly introduce Spinco Biotech Workflow Solutions. As you can see from this slide, Spinco Biotech is your one-stop shop for all your solutions in upstream and downstream workflows. Beginning with discovery and development to cell line screening, we represent world-class leaders such as Shimadzu. Our association with them goes back to 38 years for their chromatography and mass spectrometry solutions, molecular devices for the microplate readers and imaging solutions, Wyeth Technology for multi-angle light scattering, Sedaria for the ELSDs, Uptech for automated peptide synthesizers, Apricon for the bioreactors, Mesoscale Discovery for the electrochemiluminescence-based bioassays, while in downstream process technology, we are proud of our association with DLAMA, the industry scale autoclaves, Temptress for the wireless temperature sensors, Lighthouse for the headspace analyzers, and SP Scientific and Biopharma for their Lyo solutions. A journey. Before I go into this next slide, we are also excited to share with you that we have a Center for Excellence for Freeze Drying at Chennai Spinco Towers. This is a collaboration between Spinco, SP Scientific, and Biopharma, offering analysis and consultancy services. So be it your thermal analysis, bespoke training courses, and also demos in case of proof of principle, we have it all under one roof. And it is indeed our pleasure to invite you all to experience this at the Center of Excellence for Freeze Drying, Chennai Spinco Towers. A journey that began in the Andes Mountains, where potatoes were squeezed to reduce the water content and taken to higher altitudes and left to freeze as a method of long-term storage, through the passage during the Second World War, when ambient temperature shipment was inevitable for penicillin and plasma to the wounded soldiers. Today, in food, in pharma, in biopharma, and vaccines, where freeze drying has come a long way as one of the critical process parameters in vaccine production, influencing potency, safety, and efficacy. Process is the product is an often repeated statement in biological workflows owing to the complexity and heterogeneity of biologics. With the race for a vaccine during this testing pandemic and freeze drying being an integral part of such workflows, we are happy to be hosting today the webinar on freeze drying of vaccines and diagnostic kits by Dr. Kevin Ward. Over to the expert.
Well, thank you very much indeed, Dr. Uma, and also to Mohan and Devansh at Spinco for the very kind invitation to give this webinar today. And welcome and good afternoon to everyone who's listening in and viewing the webinar. So it's my great pleasure to give this presentation, and hopefully everyone can see my screen. Let me just make sure I'm sharing it. So I've entitled this Freeze Drying of Vaccines and Diagnostics, Diagnostic Kits, because really this is very relevant at the moment, as Devansh was saying, in today's world. We're particularly interested, of course, in COVID-19 diagnostic kits and vaccines. But of course, what I'm going to say will equally apply to different vaccines and diagnostics. And in fact, as you will see, we'll also go into a little bit of detail on biologics and protein-based materials in general, whether they're therapeutics or vaccines or diagnostics. So a little bit about my background to start with. I originally started by doing a bachelor's in chemistry before I then embarked on a PhD in freeze drying at one of our pharmacy schools here in the UK. And that was partly sponsored by Biopharma, which then was a very young company, as you'll see in a moment. And also I had some industrial input from one of our government departments, CAMAR, at Porton Down. And so it was a sort of very nice relationship between the university and the company and one of our government departments. After doing that, I then went to work for Pfizer in the UK, where I was working mainly with small molecules, and then went back to the university to work on a government project where we were concerned with producing single-dose oral and nasal vaccines that were freeze-dried. And then one day I received a phone call from the owner of Biopharma, the founder, a man called Tony Gaster, and he invited me to come and join the laboratory, which was then very new. So I joined 20 years ago. I was the only scientist, but now we've expanded over the last 20 years. So we're a team of 9 dedicated freeze drying scientists. And actually within my team, there are 4 people who have done their PhD in freeze drying in the pharmaceutical sector. Over the last 20 years, we've worked on more than 3,000 projects for more than 600 client companies worldwide. We are always surprised at how many people are actually doing freeze drying. But with more and more discovery and development of vaccines, diagnostics and therapeutics, there's no sign of freeze drying stopping anytime soon. So I'm really glad that I'm in such a nice specialized niche sector. My day-to-day responsibilities are for overseeing all aspects of the scientific work. Most of the things we do here in our laboratory are for our customers. We have generally between 80 and 100 projects running at any one time, shared between the 9 of us. So we're a very busy laboratory. And when we get time, we also try to run our own internal research and development projects. And that over the years has included developing new instruments for us to use ourselves and for our customers to use as well. And also pushing the science of freeze drying. So looking at new applications, and some of the projects that we've had funded by the UK government and the European government have been involved with things like the freeze drying of probiotic bacteria, of collagen wound repair materials, and red blood cells. And so we've had a lot of experience in a very wide range of experience within freeze drying and the medical sector. We've also maintained a good relationship with universities here in the UK, Imperial College and University College London, and Cambridge University being among them. I also am an industrial lecturer at one of our schools of pharmacy, because we find that freeze drying is often not taught on degree courses. So it often gets missed from pharmacy, from chemical engineering, from chemistry. And so we see it partly as our role to help educate people in freeze drying. And finally, after I had been in freeze drying for 25 years, finally I managed to have time to co-edit a book, as you can see here, in conjunction with a very old colleague of mine, Dr. Paul Matichuk of our National Institute of Biological Standards and Control. So that's a little bit about me. For biopharma, there are many aspects to the biopharma group. Most of our operations selling equipment are within the UK and Ireland and France. So we do all kinds of things in terms of selling equipment, but the division I run, BTL, it's concerned with providing consultancy, instruments and training specifically within freeze drying. So really that's what my job is concerned with. And of course we've had a very long established relationship with Spinco, who've represented us in India for quite some years now. So the services that we offer: contract research and consultancy, so we can help. We always say that we're here to help people as much or as little as they need us. And certainly if you're new in freeze drying and perhaps you have a small number of formulations, maybe one product, we can just do one analysis. And of course, as you heard earlier, that can now be done in Spinco Towers in Chennai in the Centre of Excellence, because we have parallel analytical capability there and freeze drying capability as we do in the UK. We have courses and webinars, of course, like this one. This is really a taster for some of the aspects of freeze drying that we can cover, and we're always very happy to give people bespoke, customized webinars or on-site courses depending on their needs. And we have developed a number of analytical instruments over the years. Biopharma was responsible for the first commercially available freeze drying microscope back in 1998. We've come quite a way since then, as you'll see later in the presentation, and that technology has developed and become more advanced. And so we've kind of got three arms to the division BTL. So I just want to give a brief overview of today's webinar, and then we're going to pause very briefly to look at one of the polling questions that were sent out, just to give a poll of your experiences in freeze drying and what's important to you. Today we're going to look at some of the fundamentals of freeze drying as they could be applied to vaccines and diagnostic kits. We'll look at some of the challenges with formulation, with cycle development, that freeze drying cycle, that process, and looking at scale up for biological products. We can look at the characterization and how that can reduce the risk and increase your knowledge base when you're thinking of freeze drying something. And then we'll finish off by just reviewing some of the expertise that we have between all of us at Biopharma and Spinco and how that can be applied to help you with your new products, or even to look at your current ones to see where there might be room for improvement. So I'm just going to hand over to Devansh at this stage, who's going to bring up the first poll.
Yes, sir. Thank you, Dr. Ward. So we want this session to be as interactive as possible, and one of the benefits of a live webinar is the audience can really get involved. So before I launch the polling questions, just wanted to remind you, if you have any questions, please type them in the Q&A box. We'll be taking them up at the end. So here's the first question. So I'll read out the question. Which is the most challenging part in freeze drying cycle development according to your present position? The options are thermal characterization, primary drying optimization, cycle time reduction, addressing cake quality, and all of the above. So we'll have 20 more seconds to answer this question. Yes, 5 more seconds. All right. Thank you, everyone. So as you can see, 90% of the people say that all of the above are challenging parts. What do you feel, Dr. Ward?
Well, that's really interesting, and it's, I think, a good illustration of the complexity of freeze drying and how everything needs to fit together in order for you to have a really nice, stable product with all of the qualities that you'll be expecting. So I guess that's that fits in with our experience, that things are very much linked together. And characterization is important, and for different products actually the priorities can be quite different, and so what we're finding at the moment is that certainly for COVID diagnostics, the priority is to get things out to the market very rapidly, and maybe the optimization, fine-tuning of the process is maybe not so important, and maybe long-term stability is not so important. Maybe even for some of these products, the appearance of the product isn't 100% important. It's actually more important to get the numbers out there. Whereas if you're producing something which is maybe a biological vaccine, and you want it to be stable for a very long time, your priorities may well be slightly different. So it's very interesting. Well, you'll be glad to know that we're going to cover all of these aspects in some level of detail or another during the balance of this presentation. So thank you very much for that input. That's very interesting. So I'm just going to talk about some of the very basics of freeze drying, just to get everyone onto the same page. The term freeze drying is quite descriptive, really, in that it suggests that we take something and then freeze it, and then we dry it from the frozen state. And so it's almost a self-explanatory term. You may have also heard the term lyophilization, and that essentially means to make something into a lyophile. So if something's lyophilic, it's attracted to solvent. Just as something, if it's hydrophilic or hygroscopic, it will attract water. Lyophile is a more general term, so it could refer to water, but it could also refer to other solvents. And I guess one of the principal features of freeze drying is that it makes something into a lyophile. So that tends to be used as a term as well. And they tend to be used as almost interchangeably to describe freeze drying. So just looking at the little cartoon at the bottom here, how does freeze drying work? Well, we'll take a liquid fill. Here we're showing it in a vial, but you'll see some examples of different formats later, particularly for diagnostic kits. We reduce the temperature of the liquid to freeze it. And then the way that we remove the ice is to sublime it. So we transfer energy into the product, we apply a vacuum, and we encourage the ice to escape as vapor and so we end up with a dried product. Now you can see that the frozen vial in the middle and the dried one on the right, the product occupies the same volume, the same amount of space in the container, and that's what we aim to do. So we aim to freeze everything. So when we freeze the product, we don't just want to form ice, we need to make sure that everything else is solidified, because the next thing we're going to do, of course, is to take the ice away. So we don't want the ice to be the only thing supporting the frozen structure. And this is one of the challenges in freeze drying. But hopefully we'll end up with a dried product that has a nice high surface area, it's nice and porous, and hopefully then it will reconstitute, rehydrate, go back into solution or suspension very quickly. Can anything be freeze dried? Well, actually most aqueous products can be freeze dried, but that does come with some caveats, as we'll see in a few moments. It does depend somewhat on the formulation and its critical temperature. We even find that we can freeze dry things containing some organic solvents. So these might be trace amounts of organic solvents, but actually you will find that certain drugs on the market, certainly quite hydrophobic lipophilic drugs, and cytotoxic drugs will be a classic example of this. They will often be freeze dried from organic solvents or mixtures of water and organic solvents. Now, as long as the organic solvents are reasonably easy to freeze in our normal freeze dryer temperature, working temperature range, and they're volatile enough so that we don't have to go to very, very low pressures, then we can freeze dry them. There might be some safety issues, there might be some other issues we need to take into account, but generally it's quite possible. One thing that we've found does not freeze dry quite so well are things like oily liquids, or those that are very highly concentrated. Sorry, I'll just go back to this slide. And one of the examples here is we often see small amounts of glycerol present in diagnostic kits. We know that glycerol doesn't necessarily freeze particularly easily in a regular freeze dryer, and this provides us with somewhat of a challenge. So we'll see some examples of that later on. Also very concentrated solutions. So once you start going above, say, 15–20% solid weight in your solution, sometimes we find that the viscosity becomes quite high, and so the ice crystals find it very difficult to grow, and then the ice will not really be able to escape as vapor particularly easily. A good example of this, an analogy of this in real life, is to consider things like a frozen syrup. You can imagine that freezing a syrup is not particularly easy because the water gets trapped. And a lot of work that we do at Biopharma involves trying to find the best combination of formulation, concentration, and container type. Now, an everyday example of where we've had to look at concentration is that we were invited at one point to freeze dry honey, Manuka honey, which we know has good therapeutic properties. But of course, standard honey almost universally will contain somewhere between 18 and 19% water. Now, of course, you can imagine that freeze drying it is virtually impossible, and slightly counterintuitively, you actually have to dilute it with water to be able to freeze dry it. And so sometimes we have to take what seems to be quite a strange approach to get something to work, but that's the kind of thing that we're very accustomed to doing here at Biopharma with our experience. So just comparing freeze drying against other drying methods, which might be a very natural thing to do, I guess the disadvantage is everyone would freely admit that it's in the cost of procuring the equipment and running the equipment, the energy costs, maintaining sterile conditions in something which is a vacuum vessel, and compared to traditional drying methods like spray drying, oven drying, fluidized bed drying, we're talking generally days rather than hours or minutes. And of course, the longer you process something, you could argue that there is a a greater risk of something going wrong. So they are the disadvantages. However, we find that because freeze drying is really developed largely through the pharmaceutical sector, it tends to be a very well controlled and very well documented process, and we can achieve very low levels of water in the product in a very controlled way. And this can provide for long shelf life, because with many products, the reason we're drying is because water is a problem, and hydrolytic reactions are often the basis for degradation. And so if we can achieve very low levels of water, then we have a long shelf life. We could also consider it a gentle method compared with the other drying methods, which often use elevated temperatures. Well, here in freeze drying, of course, we're starting off by removing heat from the product. And so even if we consider traditional Arrhenius kinetics or Williams-Landau-Ferry kinetics of degradation, the first thing we do is to remove heat. And so that in itself can be a good thing for stabilizing liquid products. When we freeze, we can help maintain the structure as well as the activity. And we'll look later in the presentation, we'll look at some of the challenges when we're dealing with proteins, enzymes, antibodies, biological molecules, and even live vaccines, that actually controlling freezing can be a real key to success. And what we hope is that, as I said a little bit earlier, if we've got a nice porous product with a high surface area, the anticipation is that it should easily reconstitute, rehydrate into the state it was in immediately before we freeze dried it. And of course, compared with other things like powder filling and powder mixing with traditional drying methods, here we can fill the liquid dose into the final container, which means that we can have a very good dosing accuracy. And of course, it's compatible with sterile manufacturing because we can apply sterile filtration in line. So there are some quite significant advantages over other methods. So we're just going to look at some of the different aspects of the stages of or phases of freeze drying, and what we're trying to do, and what kind of science we can apply. So we can start with the freezing process. So as I said, we want to freeze the solvent, usually water, but we also need to freeze the solute, because the next thing we're going to do is to take the solvent away. So we want to immobilize ideally everything. And at the end of the freezing process, we should have a defined structure that's ready for drying and hopefully optimized for drying. Now, in order to do that successfully, we need to take into account during each stage of the process various critical temperatures. And these can vary depending on the type of product, the type of formulation, ingredients that we have. So in the frozen state, we're primarily concerned with the frozen state glass transition temperature for amorphous materials. Now, of course, most of the biological materials we will encounter will be at least largely amorphous, at least partially amorphous. So if we've got proteins, sugars, antibodies, polymers, and other stabilizers, maybe amino acids, then these will typically be amorphous. And so we will be concerned with the glass transition. For purely crystalline materials, and in this application, it's very unusual that we've got pure crystalline materials. It usually is more applied to small molecule drugs, but there we'll be concerned with eutectic temperatures. We may have components in our formulation which could crystallize, and we may want to control that. In which case we can ascertain the crystallization conditions to optimize the frozen state structure. And then we come to primary drying. So we divide drying into two segments. The first is primary drying, which is where we remove the freezable water. Now that in itself might be a surprise to find that maybe not all of the molecules of water that were in our in our liquid formulation might be freezable, but most of them will be, and we will remove those by a process of sublimation, where we go directly from solid to vapor without passing through the liquid phase. And here we're primarily concerned with the critical temperatures that are the collapse temperature for amorphous or partially amorphous formulations. It will be the eutectic temperature for most crystalline formulations. And for mixtures of formulations which contain both crystalline and amorphous components, we might also be mindful of something that we call microcollapse, which we'll look at a little bit later on. And then finally, we're interested in secondary drying. Well, you remember that some of the water will remain unfrozen, and examples of unfrozen water are hydration cells of proteins, or enzymes or antibodies, or waters of crystallization of salts or sugars, and we might want to remove some or most of those molecules of water by desorption in order to achieve our low level of moisture that's going to hopefully give us our good long-term stability. And here in secondary drying, even though we've removed all the ice, it doesn't mean that it's safe to go maybe rapidly to room temperature or elevated temperatures and then take the material out of the freeze dryer straight away. We might need to be careful about how quickly or slowly we raise the temperature. And here we'll be mindful of what we call the dry state glass transition temperature, and that's called TG. So you might notice that the frozen state glass transition is TG prime, and the dry state glass transition is TG. And these are things that we can characterize using some of the methods that we have available. Now here I'm just going to pause again for a moment while we go to the next polling question, which is regarding some of the methods that you might use to characterize your products.
Yes, sir. Thank you, Dr. Watt. The next question is coming up. Yes. So the question is, I'll read it out. Your current thermal characterization method includes DSC only, freeze drying microscopy only, both FDM and impedance, all of the above, or none of the above. So we have 15 more seconds to answer. All right, 10 more seconds.
And it's a request, please do not post your answers in the chat box. You need to tick the right answers in the polling question.
All right, I'm ending the question. Thank you very much for your answers. So again, this is more of a mixed crowd. So 43% believe that, their current methods include all of the above. And 15% have DSC only, 13% have freeze drying microscopy only, 25% have both FDM and impedance, and 5% have none of the above. Okay, wow. Right, thank you everyone.
Well, that's excellent. Thank you very much, and it's very interesting, and I think a measure of how far freeze drying related analysis has come in the last 20 years or so. Because I think if we'd asked this question 20 years ago, we might have some people saying that they just use DSC, but that would probably be pretty much it. So it's extremely impressive that so many people are now applying so many characterization methods to their formulations. So it's really excellent. Thank you very much for that. That's really interesting result. So I'm just going to talk about some of these methods and how they can help us reduce risk in freeze drying. And actually this will apply for any product that we're considering freeze drying, but not least vaccines and diagnostic kits. And so freeze drying microscopy is, as you would expect from the name, the study of freeze drying at the microscopic level. And this allows us to look at various visual aspects of how a formulation or candidate formulation behaves at different temperatures, and so we can get a good idea of the conditions that we would need to use in the freeze dryer to process it successfully. And effectively, a freeze drying microscope is a mini freeze dryer or a micro freeze dryer. And here we have our freeze drying stage, which is on the left-hand diagram. It sits onto the platform of a microscope or an imaging station, and we can view the sample from above. And on the right, it shows the inside of the chamber in more detail. We can see that it centers around this cylindrical silver block in the center, which is effectively like a small freeze dryer shelf, if you like. And this is controlled by a cooling and heating mechanism. The cooling is through liquid nitrogen, which comes through this tube on the right, insulated tube from a dewar, and it gets circulated through the block and then out the other side. We also have a heating element, so these two arms which come down from above the stage are part of a heating element, and the flexible wires that you can see that go in to the left of the stage platform are a PT100 resistance thermometer, and that helps us measure the temperature of the stage very accurately. It's not a new idea. In fact, Professors Alan McKenzie, who is in Washington State University in the USA, and Louis Ray, who was the director of R&D for Nestle for about 20 years after his PhD, which was in cryomicroscopy. And actually, Louis Ray was one of the first people who used the word lyophilization, and he was also responsible for accelerating the freeze drying process for coffee. So they had, both of these people had quite different backgrounds, but they were working on the first designs of freeze drying microscopes back in the early 1960s. But it was only really when we came up with our Lyostat 1, as we now call it, that things really became more readily available. And here's just a gallery of images that we can expect to see. This is a very simple example, sucrose, and actually many biological formulations are sucrose based, because sucrose is a non-reducing sugar, and we know that it can freeze dry very well. In fact, we know from some of the later studies that we've carried out using our micropress instrument, where we've looked at the mechanical properties, sucrose tends to have very good physical characteristics. So here on the top left, we can see our liquid sample that we've put onto that silver block, and you can see the circle around it is the aperture in the block that you saw on the previous diagram. So the small hole in the center of the block is what we're now looking through. The top middle photo shows our frozen sucrose solution. And we're using what we call a first order red filter in order to artificially color the ice crystals according to the direction that they're facing. And you can see in the third picture on the top right, we've just applied a vacuum, and our sample is now freeze drying from the edge of the sample inwards, and we've got a band of dried structure that has been created. Here we're at about -40 degrees, so we're below the collapse temperature of sucrose, so everything looks fine. And I'm going to jump to the picture at the bottom middle, and you can see that after having raised the temperature, behind the drying front, so between the frozen material and the dried material, we've now induced collapse. And what we can do is we can pinpoint the temperature at which the first defect started by rewinding through our gallery of images. And actually now, if we go to the picture on the bottom left, you might just be able to make out the first imperfections. I'm not sure whether you can see the cursor on the screen here, Just behind the drying front towards the center, we have some very, very small holes appearing in our sucrose sample. And so this we might identify as being the first point of collapse, or the onset of collapse. And then as we raise the temperature further, you can see on the bottom right, we've lost all of the structure behind the entire drying front. And actually now we have literally got syrup as we're freeze drying the sucrose above its collapse temperature. So we can pinpoint this very accurately under the freeze drying microscope, and that's arguably the first piece of very useful, important information when you're about to consider freeze drying the material and trying to figure out what temperatures to use in your freeze dryer. Now, freeze drying microscopy, we tend to say that it gives us almost everything we need to know about almost all formulations, but there are a certain percentage of formulations which have much more complex behavior, and several examples where the active ingredient is perhaps accompanied by I wouldn't say a cocktail of different ingredients, but perhaps certainly a number of different ingredients in the formulation, and therefore what you see under the microscope might give you a good indication of the temperature at which the material will physically lose structure and have visible defects after freeze drying, but it might not necessarily tell you everything you need to know. And this is where our impedance analysis comes in. And again, this is one of the first things we did when we were a very young company. Biopharma developed what we call a resistance analyzer, Liotherm. And this was very simplistic technology. And what we simply did is to put our formulation into a glass vial. We inserted some resistance probes, which measured the resistance with a very low voltage AC circuit. And what would happen is that when we froze the solution to very low temperatures, we saw complete resistance, in other words, no electrons flowing at all. And we would then slowly warm it. And usually before we expected there to be any mobility, we would start to see the resistance reducing. What we found, however, was that when more biological materials were freeze-dried, so containing proteins, polymers, antibodies, enzymes, and maybe not so many salts, we found that traditional resistance analysis did not always give us a very detailed information as to what was happening. And so actually, by pure coincidence, about 20 years ago, I was at a freeze-drying conference where we had a tabletop exhibition with our simple Liotherm instrument, our resistance analyzer, and who should walk past our exhibit but Professor Louis Ray, so one of the inventors of the freeze-drying microscope and the R&D director at Nestle, and he said, I've got a better idea than this. Instead of using resistance, which only tends to be very good for very polar and very ionic molecules, so small drug molecules would be a classic example of something where resistance would work. He had found that by looking at impedance instead, that will give far more detailed information as to what would be happening in more complex formulations, which maybe contained more biological molecules. And so actually we worked with Professor Louis Ray. He very kindly collaborated with us to produce Liotherm 2 about 14 years ago, and we've since developed Liotherm 3, which has slightly expanded impedance range. And so what this allows us to do is to look at the molecular mobility in our frozen solution. And actually we combine that with differential thermal analysis. And so what this instrument does is it measures the impedance. And here on the bottom left, we have a graph of cell culture medium. Now, this is a classic example of a real cocktail of many, many different ingredients. You can imagine that cell culture medium contains glucose, amino acid, pH stabilizers, sort of pH indicators, colored pH indicators, and various other things. And it can be quite difficult to understand everything that's happening that's relevant to freeze drying. But what this graph shows is the impedance is shown in pink, and this is on the logarithmic scale on the left. And at low temperatures, the impedance is very high. As we warm the solution from -100, we can see various changes culminating in this rather large change starting at -45 degrees, where the impedance drops, and it's dropping logarithmically from more than 1 mega ohm to virtually nothing. And we combine that with differential thermal analysis. So the red line here is showing us as we warm the solution from -100, we see a slight exotherm around -70. Well, that coincides very nicely with the change in the impedance. And that indicates that somewhere around -75, the frozen solution starts to become slightly more mobile, and that is just ahead of this crystallization. So they fit together very nicely. But in DTA thermal analysis, we didn't really see any other major thermal events until we got to around -23. Well, we know that there's sodium chloride in cell culture medium, and we know that that melts around -21, but in this case, it's going to happen slightly earlier because we have other things in our formulation. But we didn't really see anything else. And so the impedance line, the change at -45, gave us an indication of mobility changes which were not able to be picked up in DTA or indeed in this case in DSC. When we looked at it under the freeze-drying microscope, we found that the cell culture medium here collapsed around -43. So whatever the impedance is telling us here in terms of the change in mobility, in this case can be directly related to the collapse of the product. What we might also be interested in is, is it worth using annealing? You might have heard the term annealing, and this tends to happen during the freezing phase of freeze drying. So annealing involves taking the frozen material and warming it to encourage things to become more thermally stable, or perhaps to encourage the ice crystals to grow. And so the impedance can give us an indication of what temperature, if we go above that temperature, where we're going to see increased mobility and hopefully more accelerated changes and more effective annealing. But also with biological products, so particularly diagnostics and vaccines, where we have quite a number of different formulation components, microcollapse could be a potential risk. Now, microcollapse will happen in solutions which typically contain ingredients that phase separate. And so what happens when we freeze something? Firstly, we see the ice crystals form, and then if we've got crystallizing components in our formulation, hopefully we'll see those crystallize out, but they will leave the amorphous components in a separate phase. And so at the end of freezing, we will have ice and a crystalline phase and an amorphous phase. Now, the crystalline and amorphous phases, when we then come to carry out primary drying, will effectively behave as two independent phases, even though they're coexisting within the same space. And so a good example of this is if we put mannitol into a formulation, which when it crystallizes has a very high eutectic temperature, but say we have glucose in our formulation, which we know will collapse somewhere around -40 or slightly below, then if we process them at a temperature which is between those two critical temperatures, so say -20, we might expect the mannitol to be fine, but we might expect the glucose to collapse. Now, outwardly, we might then have a product which looks like a very nice freeze-dried product, but if we looked at it under the microscope, we would find that the glucose might be collapsed, dense, and harboring moisture. So this might be something that we need to address for complex formulations. And this type of analysis is a classic example of something which can indicate when one phase is becoming softer, even if the other one doesn't seem to be changing visibly. So once we've got some basic information about our formulation, we then need to consider how are we going to apply that knowledge when we come to freeze dry that formulation. And so here we have an example of a freeze dryer, and it has a number of shelves, and those stainless steel shelves are controlled in terms of their temperature by a fluid which circulates through the shelves, in this case a low viscosity silicon oil. And therefore, in our freeze drying cycle, we can set the shelf temperature, and we can expect that to be maintained throughout the process. So this is one of our control parameters, and the other control parameter is chamber pressure. So these are the things that we directly control in this freeze dryer. We might also be interested in the condenser temperature. So on the bottom right of the freeze dryer, this chamber here, the cylindrical smooth wall chamber is our condenser. So that's where we're going to deposit the vapor that's coming off of the product. Now, the one thing we need to make sure is that this is below the temperature of the product in order to attract the water to it. And so we traditionally cannot control the condenser temperature. We can't dial in a set point, but it typically will be maintained at its lowest possible point throughout the cycle, but we typically monitor it in order to make sure that everything is running well. We're also interested in what's happening to the product, and this is where our collapse or micro collapse or eutectic temperature can come into play, because what we're ultimately interested in is what is happening to the product when we use these freeze drying conditions. And so we can classically use thermocouples or resistance thermometers in our freeze dryer in sample containers of our formulation to see what it's experiencing in terms of its time temperature profile. And so if we have a collapse temperature of -20, you can see that in primary drying, all of these product temperature probes are below -20 until they reach the point where they start to rise partway through primary drying, indicating that those samples are becoming dry. And this is so how we use the information. On the left-hand side, on the freeze dryer itself, in the top of the chamber on the left, through the perspex door, you can see that there are these thermocouple wires that are plugged into this bank of sockets at the top. And these are the thermocouples that we will put into our product to determine what is happening. So what happens if we exceed the critical temperature? Well, classically, we know that, as we heard earlier from Dr. Uma, some of the large leaps in freeze drying happened in the 1950s and 1960s for medical products. And we know that in those days, people would not have been looking at the collapse temperature, and there would have been a great deal of trial and error in selecting their conditions. And probably one of the things people experienced, a part of that trial and error development process would be things like eutectic melting of crystalline materials. And so here on the top left, this vial is showing a solution which we know is a crystallizing solution, but it has a very low eutectic temperature. And so when you go above that temperature in freeze drying, the ice may remain solid, but between the ice crystals, we will have a eutectic liquid. Now, what does the liquid do under vacuum? Well, it could then start to boil, which is what's happened in this case. And so you can see that the product has effectively erupted up the size of the container. It's almost managed to escape, in fact, through the stopper, which would have been open during the sublimation. So this is, of course, a risk. Over on the right here, these are examples of amorphous materials. The top one is a sample which has just started to lose its structure. Perhaps not significantly, but the indication that it started to lose its structure here comes from the fact that it's a very uneven shape. It's no longer uniform, it's not cylindrical, the top isn't even horizontal. So this is an indication that we've just started to see softening and loss of the structure in this particular example. So if we saw this, then the next time we freeze dry this material, we'd probably know that we'd been close to its collapse temperature, and so we might just need to use slightly less energetic conditions next time. The vial below it, sorry, let's go back. The vial below it here, you can see that the product is now completely at the bottom of the container. Now this is glucose that I mentioned before, so it will collapse just below -40. So if you exceed that temperature, all of your glucose will end up effectively as a syrup at the bottom of the container. Now, believe it or not, all of these vials, these two vials on the right contain exactly the same amount of material, about 100 milligrams. But you can see that at the bottom here, it's completely collapsed. So it has virtually no surface area, maybe a couple of square centimeters, whereas hopefully the one at the top, well, we might be talking in terms of square meters per gram. So it's actually quite significant. So it can be a very big difference. Well, you might say, well, you know, for my product, maybe appearance is not particularly important. Well, that might be true, but often what we find is that if something's lost structure, then that tends to bring with it other problems. And so the moisture level can be very high. So in this glucose sample, you can probably appreciate that we're not down to very, very low levels of water. You know, it effectively looks like a syrup, so maybe it's 10% water, possibly even more. Well, that's probably not going to be stable for a very long time. If it's got poor stability, then that could also mean that the molecular mobility is higher, and so there could be other interactions happening. And low surface area means it's going to be difficult to reconstitute. So no, appearance is not everything, but we do tend to find that if the appearance is bad, then other problems seem to follow. So just a few slides on some of the very specific elements of vaccines and diagnostics that we might be interested in. The types of vaccine that are freeze-dried, well, it can range from anything from recombinant proteins through DNA or RNA-based vaccines, through inactivated organisms, right through to live vaccines. Now, depending on the kind of vaccine we're processing, our priorities and our expectations of the product might be slightly different, and there are different levels of complexity, of course, if you're comparing a simple protein to a live viral vaccine. Now, what we tend to find is that we have to use some kind of stabilizers. We might also see these in conjunction with vaccine delivery systems or controlled release systems, or some sort of particles, like microspheres, biodegradable microspheres, which could be an adjuvant as much as anything else. But what we also find is that traditional adjuvants, and particularly the aluminium salts, don't tend to freeze dry very well. So almost inevitably, these will be used to add to the lyophilized components before administering to the patient, rather than being there before freeze drying. There hasn't been a great deal of success freeze drying to classic adjuvants over the years. On the other hand, diagnostics, well, we don't tend to have living diagnostics. We go right down to very small molecules. So examples here would be test reagents for water contamination, or drug detection kits, which might be carried out in a mobile system, either at the roadside or airports, for example. Now, small reactive molecules, again, the reactivity of the molecules are what makes them good at being diagnostic reagents, but of course it also means that they tend to be reactive and not very stable. And so usually the primary concern here is to take as much water out as possible to reduce the speed of degradation kinetics, and sort of stop or slow down any hydrolytic reactions. And these tend to be quite simple, as long as you don't degrade the small molecule or change it to a completely different molecule, generally that's fine. What we tend to be more concerned with these days, or what we see more of, are the biomolecule-based diagnostics. So in this case, and again, a good recent example, current example, COVID diagnostics. And so we typically see ELISA tests where we have the enzyme-linked immunosorbent assay, where quite often it might be the enzyme or the antibody that needs to be stabilized by freeze drying. Or in terms of the PCR mixes, so the polymerase chain reaction DNA-based diagnostics, it tends to be enzymes such as Taq polymerase that are the limiting factor to stability, and that we need to use freeze drying to stabilize them. Again, the primary concern is to remove water, but also ensuring that activity is retained throughout freeze drying and after freeze drying. Now, one of the challenges here is that if we compare these to small molecules, where all we want to do is to maintain the chemistry of the molecule, here we need to be more careful, because it's not just chemical degradation that's going to lead to inactivation of these molecules, but sometimes even a subtle change in the three-dimensional shape of the molecule. So if the molecule twists or becomes distorted, that can be sufficient to make it inactive. So we might not have chemically changed it, but even if it changes subtly, that can cause problems. And so that applies to enzymes and antibodies and various other types of protein as well. Now, we can come up with some general rules. In fact, we have done over the years. We've come up with some general guidelines as to what to use and maybe what not to use in terms of formulation ingredients. But it's still very much the case that different biological molecules have slightly different destabilization mechanisms, and therefore, even if two protein molecules look very similar, it doesn't mean to say that they're going to have the same kind of sensitivity or robustness to processing or storage conditions. And so we need to look at them on a case-by-case basis. And what we've found historically is that it's always best to look at the formulation, the freezing, the drying, the container, the format, and the concentration together holistically, and that tends to give us the best outcome. Now, if we take that one step further and we've got a live viral vaccine or a sort of probiotic, and we want to maintain viability, so when we freeze dry it and reconstitute it, we want it to kind of spring back into life and show viability, then there's an extra layer of complexity involved, of course. And again, we need to assess what are the potential issues that could destabilize it in freezing, what about drying, what about long-term storage, and actually, is there any aspect of rehydration that's important? Sometimes just adding water, cold water to something is not always the best way to rehydrate it if it's a living material. So people have looked in quite some detail at how perhaps this might need optimization. So just to take some of those things into account, and particularly focusing on freeze drying and the freezing and the drying, it's long been established that freeze drying can actually cause problems for biologicals, and there are some stresses associated with freezing and with drying. And we freely admit this, although we typically consider freeze drying still to be less of a stress than some of the more traditional higher temperature drying methods. And usually we've been able to find a way around these stresses, either by adjusting the freeze drying conditions or adjusting the formulation ingredients, or a mixture of both. So a good example here of freezing being destabilization, being a potential source of destabilization, is what we call the freeze concentration effect. And now this will always happen in freezing. So the first thing that happens is that the ice crystals start to form. And actually what we're seeing initially is the formation of pure ice. And so the ice grows throughout the solution. And what it does is it forces everything else to become more and more concentrated between the ice crystals. So our active ingredient or organism will be forced into a smaller space. Molecules will be forced to interact with each other where perhaps they didn't in dilute solution. There might be osmotic effects on cells, and there can sometimes be a pH shift on freezing, which can be notoriously difficult to measure. So if we have something which falls into the category of being very sensitive to freeze concentration, then that might direct us to perhaps use very rapid freezing conditions or to use more stabilizers. On the other hand, we see that some proteins are susceptible to what we call interfacial effects. So they may not be destabilized by freeze concentration, but they might reside at the ice-water interface. And we know that's a dynamic microenvironment. And we know that if proteins are damaged at that interface, then the damage tends to be proportional to the surface area. And so again, that might direct us to change our freeze-drying conditions and maybe use slower cooling. Again, maybe we need to reconsider the formulation in that case. So that's a couple of examples of how freezing can be destabilizing. And then, of course, we're going to start to remove the water. We're going to remove the ice, but we might also remove structural water, as we might refer to it. And so looking at the 3D structure of the protein or enzyme or antibody, we can find that perhaps in order to maintain the 3D shape, there are strategically placed water molecules that are partly responsible for holding that 3D shape. Now, if we're risking taking them away by freeze drying, then maybe that's going to have a disruptive effect on the molecule. So this is typically where we use our lyoprotectants. And so at the bottom, as you can see here, we're going to need to adapt our freeze drying strategy and even our formulation strategy according to the sensitivities of the molecule or the active material in question. So different freezing conditions, maybe controlled nucleation, which we know is available commercially on some of the SP Scientific freeze dryers now, or the use of annealing to try to make things more thermally stable, and maybe to adjust the formulation. So everything, as much as we have some good knowledge over the last several years on the freeze drying of biologicals, we still need to approach them on a case-by-case basis. And just to look at some of the additional issues with diagnostics. So some of the issues with diagnostics is that they often are lower volume compared with vaccines. So with vaccines, usually between 500 microliters and several milliliters. Well, with some of the diagnostics, there might be tens of microliters. And so dispensing accuracy, avoiding evaporation before you've started freeze drying can be an issue. They can be, some of the components can be supplied in the presence of glycerol-rich solutions. There can be sealing issues if they're not in vials, and there can be moisture uptake from or through packaging. And what we've found, particularly in some of the diagnostics we've worked with over the years, is that we've had to be very careful about the packaging. If we've got a successfully freeze-dried material, it's probably quite hygroscopic. But of course, if they're very low mass, then that means that even a small amount of water going into the product over time could represent a very high percentage of the product mass. So immediately after freeze drying, we might have 2% moisture, but over a period of months, we might find that suddenly that goes up to 10%. We know that plasticware tends to contain moisture at the beginning of freeze drying, which doesn't always come out during freeze drying, and so we have to then optimize the secondary packaging and use things like desiccant sachets in the secondary packaging with our, in this case, a sealed 96 well plate. So you can see in the top left, we spent on this project quite a lot of time optimizing our sealing conditions in terms of the time and temperature for the heat seal, only to find that actually the 96 well plate itself was much more permeable. The plastic was much more permeable than our really good seal. And what we found was that plates that are even made from the same polymer, like polypropylene, polyethylene, they can have different porosities from different manufacturers. And so this became a problem that we actually didn't expect. So sealing is really important to optimize, and we typically use a nitrogen dry box, as you can see here, a nitrogen tent, where we can limit the humidity and limit the amount of oxygen while we carry out the heat sealing and we carry out the secondary packaging. We also use this kind of nitrogen tent to sample for moisture analysis and DSC and the other things that we might apply after freeze drying. If we've got small cakes or small beads, small freeze dried materials, then we often have to combine multiple samples. So it's always best to manipulate them in a controlled environment so that you don't subject them to uptake in atmospheric moisture. And again, we double wrap things with desiccant in this environment. And so just to finish off by talking about some of the challenges of the freeze drying process, we know that what we've often encountered for diagnostics is that they often have very low collapse temperatures. And looking at our phase diagram here, we can see that sublimation has to happen. If we want to go directly from solid to vapor, we're talking about relatively low temperatures. But what we also then have to do is to adopt very low pressures. So the laws of physics tell us that if we've got a very low temperature, then in freeze drying we have to use a correspondingly low pressure. So this pressure, the pressure will be related to this line here that divides the solid and the vapor states. So if we're just below 0 degrees, then probably anything just below 6 millibar will be fine. But for most products, we need to go way lower in temperature, and so we have to go correspondingly lower in pressure. Now, one of the issues with that is that in freeze drying, we do rely on getting heat into the product to keep drying it. And we've got radiative heating, we've got direct conduction, but we've also got convection. And convection is also termed gaseous conduction. And as you can expect, gaseous conduction relies on there being a certain pressure in our freeze drying chamber in order to effect that heat transfer into the product. Otherwise, we're going to have extremely slow freeze drying. Now, if we've got very little convection because we're using very low pressures, now 40 microbar is typically considered a pretty low pressure in freeze drying, then we're going to be very reliant on radiative heating and conduction. Now, one of the issues with that, it might not be a challenge for your freeze dryer to achieve those low temperature, low pressure conditions, but what it will mean is that your freeze drying cycle becomes much more reliant on the design of the freeze dryer itself. And therefore what you can find is the shelf spacing becomes critical, and that's something that you can't always control in a freeze dryer. It's sometimes a feature of the design. And so when you go to a different freeze dryer and apply the same freeze drying cycle with your same types of container, that you have to make quite significant adjustments in your scale-up process. I guess the plus side is that that's something that we're extremely familiar with dealing with here, but it makes it more of a challenge. So just before we go into the very final section, which is actually just a case study, I think we've got another poll question.
Yes, thank you, Dr. Watt. So another question coming up. So I'll read out the question. What are the typical issues that you're facing when scaling up your freeze drying process? The options are vial breakage, partial meltback, micro collapse, reconstitution properties, and addressing cake quality. I repeat, what are the typical issues that you're facing when scaling up your freeze drying process? Vial breakage, partial meltback, micro collapse, reconstitute properties, and addressing cake quality. So 10 more seconds for this answer. All right, thank you very much for your answers. Yes, sir. So what is your opinion on this, Dr. Watt?
See, I can't see the
yes, Dr. Watt.
Yeah, thank you. Yeah, so addressing cake quality wins. Yeah, I mean that really, I guess that really demonstrates that cake quality is important, not just perhaps from a visual perspective, but also perhaps it sometimes goes hand in hand with some of these other issues that we talked about. So melt back and collapse and reconstitution will all be affected by cake quality, or all feed into cake quality. Vial breakage, that's also interesting. We've seen vial breakage over the years, and what it tends to be related to is the coefficient of expansion when your formulation ingredients may be crystallized. And a good example of this is if you've got relatively high percentage of mannitol in your formulation. We do know that mannitol does tend to expand. And what we can then perhaps do is to try to make sure that happens before we start drying, and before we build up that dry crust on top, because sometimes then the crust means that the expansion can't happen upwards, and so it has to happen downwards and outwards. And sometimes the vial is not as strong as the ice, which seems quite strange to think of that, but we know that ice is extremely strong, and so that can be why that happens. Micro collapse, that's really interesting. 33% say micro collapse. So again, that's where the lithium, the impedance analysis can be really helpful in showing you what is the lower of the critical temperatures associated with either the crystalline or amorphous part of the formulation. So that can give you an idea of what is a limiting factor that governs the I guess the maximum product temperature, the critical temperature that you need to think about. Fewer people having issues with reconstitution, but again, that can sometimes be affected by the cake itself, and of course that's highly dependent on the active ingredient as well. So very interesting that cake quality is a pressing issue, and actually, as I mentioned, one of the instruments we've developed much more recently is the micropress, which is a sort of a structural mechanical testing device for freeze-dried materials, and it's actually designed to look at them in situ in the vial or in whatever container that they're produced in. So we don't have to take a sample physically and use an offline method to look at the structure and the Young's modulus and the strength. We can do it in situ. So we are trying to do our bit in making certain aspects like this more quantifiable for quality purposes. So just to go on to the case study, so this is from a biological product that we worked with a few years ago, and our customer approached us and said they've got a very long freeze-drying cycle, and they were keeping the product safely below -40, having established that the collapse was just above -40, and therefore they had quite a long cycle. So the first thing that we did is to say, well, let's reanalyze the formulation. And what we found was that in the initial determination, that the formulation had not been annealed, so the annealed form had not been investigated for collapse. Whereas actually when we annealed it, we found that the collapse temperature was then -25.6 rather than -39.6. So actually annealing it meant that the collapse temperature increased by 14 degrees. Now, as a rule of thumb, if you have something which is, say, 10 degrees higher in collapse temperature, then generally speaking, you should be able to roughly double the freeze drying speed, the speed of sublimation or primary drying. We carried out impedance analysis, which also suggested that once the material had been annealed, it had a eutectic melt that was somewhere just above -27 and finishing at -21. Well, actually, there was sodium chloride in this formulation, so perhaps it wasn't a huge surprise, but maybe without annealing, the sodium chloride was finding it difficult to crystallize. Just confirming that with DSC, so here we saw a eutectic at -23. So as you can see in the blue trace here, we've got this slight dip in the line just below -20. So that confirmed what we'd seen. And then when we looked at the customer's original cycle, you can see that in order to make sure that they can guarantee to keep the product below -40. They'd used a shelf temperature of -45 quite consistently throughout primary drying, as well as in freezing. But you can see here there's lots of 20-hour steps. So these 1200-minute steps here on the right, they're all 20-hour steps. So that actually resulted in a freeze drying cycle that was 149 hours. Actually, the product didn't look too bad. It's in this in these sort of relatively short molded vials with quite a high neck size. So, you know, actually in terms of impedance to vapor flow, it's not too bad. And the customer is achieving a relatively low moisture content, and they were very happy with that. But they weren't really happy with the length of the cycle. So it's nearly a week, it's a 6-day cycle. And so the first thing we did is to say, well, now we know that annealing the product is going to increase its collapse temperature. The first thing we're going to do is apply an annealing step. And so when we applied the annealing step, so we annealed it to -25 and kept it there for several hours to make sure that it would be more thermally stable. We then found that when we started drying it, we could use a slightly higher shelf temperature, and we can reduce the drying time at least by 1 day. So that's a good start, and still achieve the same moisture content as before. What we then did is, knowing that we'd annealed the structure and opened up the ice structure and reduced the impedance to vapor flow, we then tried using a higher shelf temperature. So we had a couple of attempts here. As you can see, we changed the conditions during the cycle according to the data that we were getting from the freeze dryer, particularly in terms of the product temperature. So still here now, it's still below about -35. Well, we'd established that we could actually have it at a higher temperature than that, but we tried to do things incrementally and step by step. We found that even by having a shelf temperature of -15 going up to -10, we could still maintain the product below its critical temperature until the product temperature started to rise, indicating that it was dry. So here now we've already halved the original drying time. So we've gone from 149 hours to 74. We've still got quite a low moisture content with good standard deviation, and the product looks fine, although there's maybe a small amount of foaming around the top here. And then in the final cycle, we just adjusted things slightly. So instead of going up incrementally, we figured that we could use a slightly higher shelf temperature early in primary drying, -10, and then we go back down to -15 when we know that the drying front is moving through the product and the drying speed is slowing down. So here it's still 74 hours like the previous one, but it's slightly more efficient at the beginning and slightly safer at the end. Slightly higher moisture content than before, but if we want to remedy that, then we will simply perhaps just need to extend secondary drying slightly to achieve a more robust cycle. So that's an example of the steps that we went through to try to improve the cycle for this product and come up with something which is more efficient. And this is the kind of thing that we do sort of pretty much all the time here. As you saw earlier, in the Centre of Excellence, we've got exactly the same kind of equipment as we have here in our UK laboratory. So we halve the cycle time simply by doing more characterization, understanding the formulation better, establishing that we don't need to keep the formulation below -40 if we anneal it. We can go to much higher temperatures, and then we explored various efficiency measures, including looking at the shelf temperature at the time of each step, the use of annealing, we achieved a much shorter cycle time, but still with an average of 1.7% moisture. And again, as I said, this is the sort of things we do all the time. Between biopharma, well, biopharma's been going over 30 years now. I've been here 20 years, and members of my team and I think together we've got way more than 50 years of experience in working with the products. And of course, then when we add that to all of the expertise that's resident at Spinco, then that's a huge number of decades of experience between us with helping our customers with their formulation, with their process in freeze drying. All of the instruments that I've talked about are things that we've developed here at Biopharma, and we use them on a daily basis in our own laboratory. But as you can see, the Spinco SP Biopharma Centre of Excellence in Chennai actually got exactly the same instruments for formulation characterization. So the idea of the Centre of Excellence was that we would set up effectively a parallel lab, and so that we can help each other. And it also means that you won't necessarily need to ship your formulations to the UK with all the current COVID restrictions on shipping things. Then this will of course also help. And between us, of course, because Biopharma has sold freeze dryers in the UK for 30 years, and Spinco has been doing the same in India, we all have a good working knowledge of the different scales of the freeze dryers and how they work, and the different design elements. And so I think we're uniquely positioned to help with scale up and ensuring that the scale up and tech transfer goes smoothly, and helping troubleshoot or even foresee in advance any issues that are likely to come up when you go from one freeze dryer to another, based on their particular characteristics. So with that, I think before the general question and answer session, I think we have another poll question. Is that
yes? Thank you very much, Dr. Watt, for the wonderful presentation. So we have one more poll question coming up. Yes, the question is, which one of the following approaches do you prefer for your critical life cycle development work? Number 1, availing knowledge through bespoke courses, trainings, seminars, procuring latest technology-built instruments, availing consultancy service, or opting for thermal analysis. The question is, which one of the following approaches do you prefer for your critical life cycle development work? Availing knowledge through bespoke courses, training, seminars, procuring latest technology-built instruments, availing consultancy service, and opting for thermal analysis. So we have 10 more seconds to answer. All right, thank you very much for your answers. So again, this is more of a mixed, these are more of mixed answers. 41% would opt for thermal analysis, 27% would opt for availing knowledge through courses, 23% for procuring latest instruments, and 14% for consultancy services. Great.
Yeah, so I think yeah, I mean that adds up. I think to more than 100%, which suggests that people are some people are sort of doing multiple, having multiple approaches, and perhaps trying to have their own training, but also developing their expertise in house with some of the technologies. So again, that's very interesting. But thermal analysis again is leading the charge, and I think that again says how much freeze drying has moved on over the last 20 years, with everyone considering that some kind of characterization, some building knowledge is important, and I think we're relying less on trial and error than perhaps we used to. So that's very interesting.
All right. Thank you, Dr. Watt. So now we'll move on to the questions. So we have one question about stoppering. So
Yes.
Since now a lot of manufacturers are going for diagnostic kits, which are produced in small vials. So how well does stoppering work in small vials?
That's a really good question. One of the issues, of course, with very small vials, and I think this is something that the first time we did it wrong, we did it wrong through experience, and we only realised afterwards, was that when we had vials that were very short, and we put them into the freeze dryer through use of the removable base tray, and so the frame stays in place while you take the tray base out. We only realised partway through the cycle that actually the frame was taller than the height of the vials that they when they would have the stoppers pushed in, and so sometimes you need to have some modified frames. So SP calls them the low profile frames in order to make sure that you can stopper them. In terms of the stoppers sticking as well, I mean that can be another issue. It can be due to sometimes the stoppers themselves. Sometimes it was found in a study a number of years ago that some rubber stoppers have a complete circle on the top, which can actually promote suction. When the shelves come down and press on the rubber stoppers, they can stay in place. And so some of the stoppers now, instead of having a complete circle in the middle, which is usually for injection, it's usually to guide the end user on where to put the needle, they have an incomplete circle now, so it's sort of two almost semicircles together. And so it's been found that using that, and sometimes having some raised nodules on top of the stopper, can also prevent it from sticking. I know that people have looked at other approaches to this, like coating the underside of the shelf above, but people always have concerns over introducing something else into the freeze dryer. What we sometimes do is once we've put all the shelves together to compress all of the stoppers in, we sometimes then leave the shelves in the closed position, and then we start to release the vacuum. And what we often find then is that the stoppers don't stick. So I hope that's helpful.
Yes, definitely. So the next question is, what are the possible reasons of micro collapse in lyophilized cake and its effects on long-term stability?
Again, a great question. Thank you for that. So yeah, microcollapse, it will tend to happen when you have different phases. So when you have a mixture of crystalline and amorphous phases, then it tends to be that if you process that formulation above the critical temperature of whichever phase has the lowest critical temperature. So I used the example earlier briefly of mannitol and glucose. So if you have mannitol and it crystallizes, the eutectic temperature is about -1.4. If you also have glucose in the formulation, well, we know it collapses around -41. And so you've got quite a big difference between the thermal stabilities of the different phases within your frozen material. And so our idea would typically be, if you know that the glucose collapses at -41, even though you might not see visible defects until you go to much higher temperatures, our general recommendation is you at least consider freeze drying it below -41. You might then say, well, that's going to give me a very long cycle, so what I really want to do is process it at -21. Well, you can try both and see whether that has an effect on the stability of the dried material, maybe by putting them on accelerated stability, for example. But the impedance analysis really is what gives us a good indication of when you have that mixture, what is the lowest critical temperature. And we will see the glucose changing very rapidly in mobility when you get to around -40. So that would be the indicator that we would typically use. But yeah, the reason that it happens is that you have things which form separate phases in the frozen state, which sometimes you can avoid, sometimes you can't avoid. I hope that answers your question.
All right, thank you. So the next question is, okay, one of the participants, they develop lyophilized diagnostic kits which are fluorescence based. When they tried to develop colorimetric-based lyophilized kit, the cake was not uniform and was slightly glassy, and the dye distribution also was not uniform, but the activity was good. So, what parameters need to be considered to get a solid cake in colorimetric lyophilized cake?
Okay, excellent question. So this is I guess, an unusual example of where the product doesn't look exactly as you want it, but actually the activity is fine. And here the dye distribution not being uniform and the cake not being uniform, typically the way that we're trying to solve that is to use things like annealing. It might also be a really good candidate for controlled nucleation. And often if something doesn't look uniform, it's usually, as long as you've processed it below the critical temperature, that would then suggest that the unevenness, the lack of uniformity probably was set up during freezing. And so we would tend to then focus on the freezing to try to see whether that can make it more uniform. Some people also say that if you quench cool things, you can often get more uniformity. So I guess the example is if you flash freeze it, or quench it, you will tend to get good uniformity. On the other hand, you're likely to get very small ice crystals that are not necessarily going to be conducive to efficient sublimation. And so you might then have to perhaps then add an annealing step to try to optimize both of the aspects. So yes, it's usually the freezing where that kind of thing is set up. So hopefully that gives you a good pointer.
All right. Thank you. So the next question is, what are the main factors affecting the rate of recovery of biological activity in a lyophilized sample?
Again, that's a really good question, and I would tend to then go back to the freezing. Now, someone called Felix Franks, who some of you may have heard of, the late Felix Franks, who used to be really a specialist in water and freezing. And he was also into freeze drying, and one of his phrases was that freezing is in itself a form of drying, because all of the freezable water you're effectively removing from contact with your active ingredient. And so he said that a lot of the problems with activity, particularly for biologicals, quite often will happen during freezing. So here we would then look to perhaps use different cooling rates. And as I mentioned very briefly in one of those slides, some things can be susceptible to freeze concentration damage, in which case you might then decide to cool them more rapidly, or you might need some more protective agent, or you might need to change the buffer in case it changes pH. On the other hand, you might have a sort of biological that is surface active and actually doesn't respond very well if you cool it rapidly, so you might want to do the opposite. So again, it's very much a case-by-case basis, and so I would typically look to try to optimize the freezing. Generally speaking, if you've got protectants in there and the drying is carried out below the collapse temperature, you should generally be fine. So I'd go back to the freezing.
All right. Thank you. So we have one question for Dr. Raghuram and Mr. Mohan. So you mentioned about the Spinco Biopharma Technology Center in Chennai. So can you elaborate more on that? What kind of support do you provide for customers in India?
Hi all, this is Mohan.
Yes, sir.
Yes, Devans, thank you. And I would like to thank Dr. Kevin Ward once again. And basically, the Center of Excellence was created to help all our customers, typically a Lyo community of India, in helping them achieving the analysis services quickly. This is the number one aspect. And second thing, basically many of the people are just availing the services, but it is also a sense for them to come and see the technology and get demonstrated. So this is also serving as a demo center. So we are offering a demonstration of the complete system, what Dr. Kevin explained here, that is typically Lyostat 5 freeze drying microscopy and Lyotherm, and as well as the micropress. It's a very innovative new equipment, and as well as the Lyostat freeze dryers. And we are also offering a proof of concept trials for lyophilizer. And besides, we are jointly working with Biopharma in taking up some of the specific work related to the cycle development together with the thermal analysis. So anytime you could approach us through Biopharma or Spinco, we are open. And I think any of your questions we'll be taking. Thank you.
Right. Thank you.
Thanks, Mohan. Essentially, this is to reduce the turnaround time, and we offer both the proof of principle study, and we can also work with you and on a consultative manner suggest what is the best process suitable for the kind of product you're working on, along with experts such as Dr. Kevin, who has over 20 years of experience in this field.
All right. Thank you, ma'am. So, due to want of time, since we're already overdue and we have shot past the intended time, so we'll be taking two more questions only. We're really sorry for the other questions. We'll get back to you with answers through email. So the next question is, how do we control on blowing of the material from vial? Dr. Kevin?
Yeah, so blowout of material, yes. Yeah. Yes, in this case, I mean, it depends on when it happens. So often this is a function of the formulation, and sometimes what we find is that certain excipients that we use can be very cohesive, and other ones could be quite flyaway and powdery. And sodium chloride is a good example of something which can be quite powdery if it's used in high quantities and maybe in high proportions compared with other things. And so it might be sometimes that you might have to adjust the formulation to stop any material blowing away, or ablation is the term we tend to use for that. It can also be that sometimes it happens early during primary drying, in which case you might simply want to reduce the speed of drying. So maybe start by using slightly lower temperatures than you would otherwise do, and then ramping up. On the other hand, sometimes it can be done, sometimes it can happen during reconstitution, or if somebody removes the stopper of a vial, for example, then it can be quite flyaway. And if that's the case, we would also need to perhaps then go back to the formulation and maybe rebalance it with things which are more cohesive.
All right. Thank you, Dr. Ward. Yes. So the last question is, so as you know, there is a global pandemic going on right now. So what is Biopharma Technologies' response to the COVID-19 pandemic in terms of support and technology for vaccines and diagnostic kits?
Well, that's another excellent question. I mean, we've worked with diagnostics and vaccines for certainly the 20 years that I've been here. So we have a lot of experience in helping with those. And in fact, you can see from some of the testimonials that have kindly been supplied that are on the screen at the moment from some of our customers, that some of them are actually producing diagnostic kits. And we've been helping with the formulation and developing the cycle for those. And in fact, quite unusually, and 6 months ago, this would have been totally unexpected to us, is that actually we are involved directly in the production of COVID diagnostics for one of our customers that are being sold to the UK government and actually other governments throughout the world. So we're doing the freeze drying of some of the components, and we're doing some packaging of some of the other components. So unexpectedly, we're hands-on in production of some of the diagnostics. But aside from that, traditionally, we have been involved in a lot of vaccine and diagnostics production, as you can see here, and hopefully some of the slides in today's presentation gave people a flavour of some of the varied things that we've worked on, and it's not just small molecules in vinyl. Some of it is is quite unusual.
All right.
Yeah. Thanks, Dr. Kevin. I think I would also like to draw the attention of the audience to the testimonial by Primer Design, which sums up the answer.
Yes, yes. Thank you, Dr. Raghuram. So yes, with that, we're already 13 minutes due, and we'll close in another 2 minutes. So we're still getting a lot of questions. So highly interesting and engaging webinar. Thank you, Dr. Watt. I
Thank
now
you very much.
request Dr. Raghuram and Mr. Mohan for the vote of thanks and concluding remarks.
So first of all, I would like to thank you on behalf of our entire customers from India. Thank you, BTL Biopharma team, for particularly Dr. Kevin Ward for your time. And this has been the very right time. Actually, we have been working on this for the last 1 month. And thank you very much for putting up the right topics. That is the need of the time. And certainly, I believe this will be there will be a lot of inputs and take-home message for all the people. And apart from all the things, it is just not the end. It's only the beginning, because we are just trying to present the kind of support and all the required assistance that can be given to our customers to support their research work and scale up issues or challenges, whatsoever it is. And as said by Kevin, and we are very open. And also, I would like to thank all our team, especially Dr. Uma Raghuram, who is heading our biopharma division. And she has taken really a big initiative to jointly work with Dr. Kevin in identifying the right topic to be discussed in this platform. And also, thank you, Devansh, for all your support in getting all these things. Thank you very much. Please, Dr. Uma, the final word.
Dr. Kevin, I have been getting requests on the recording and sharing of the slides. So do let me know if that is a possibility, and then we'll have these sent out to the various guests who have registered for this webinar. Would that be a possibility, Dr. Kevin?
Yeah, I will check at this end, and we will do what we can to sort of make things available in some form or another.
Yeah, sure. Yes, I guess we come to the end of this session. It's been a very engaging session, and a pandemic notwithstanding, I'm sure we have to be in sync with the new normal. I don't think it's about fighting the COVID, but it is more about learning to live with it, and not just live, but thrive and succeed. Having said this, both Spinco and Biopharma, we are very happy to be partnering with you in your research work, be it related to COVID or any other biological assays that you'll be working upon. Feel free to reach out to us. Thanks once again to Dr. Kevin, Ms. Sally, and Ms. Clara from Biopharma. Thanks, and we are indeed very happy to have been part of this webinar. Thanks to all our guests for their patient hearing and the wonderful questions. Stay tuned. We have to get back to you on some of the questions that we could not take up during this session, but rest assured, it would be either Devansh or me who would get back to you with the answers. Thank you so much.
Thank you, Dr. Raghuram. Thank you, Mohan sir. Thank you, Dr. Ward. Thank you, Sally. Thank you, Clara. So we come up with a lot of events and activities, and follow us on social media. You can follow Biopharma and Spinco Biotech on LinkedIn to stay updated with all the recent events, all the latest events. So stay healthy, stay safe, stay connected. Thank you. Thank you very much.
Thank
Bye,
you.
everyone.
Bye.
Bye.